In this video, we will discuss about the effect of sting and BH3 mimetics on the AML patient sample. In the previous video, I have mispronounced this AML as AMC. So, in the previous video, we have understand the effect of sting and BH3 mimetics on AML cell lines. In this video, we will understand its effect on this AML patient samples. So, what it is, they did the patient-derived xenograft on the mouse models to induce the pancreatic to induce this cancers on the mice, and then they use sting and BH3 mimetics for the treatment. So they used a different AML patients samples that is genomically different. So what does they mean by genomically different is that they have some TP53 knockout samples also, and they have some TP53 wild type samples also. So TP53 knockout samples are these bottom two samples, and they did this treatment of the mice with the diazole, venetoclax, and this venetoclax plus diazole in 1:1. And what they have find out in almost all the cases, diazole shows some decrease in the tumor cells or live cells, whereas venetoclax has almost no effect or very minimum effect on the TP53 knockout cells, whereas the combined therapies, that is venetoclax plus diazoles, has a maximum impact in almost all the cell lines. So after validating that the sting agonist could trigger the cell death, and its efficiency can be improved when sting agonist can be used with this BH3 mimetics, they wanted to check this results on the mice model, humanized mice model, or we can say SG3 mice models that are in which the patient-derived xenograft has been implanted. So they use two different cases, where in one case they use the PD53 knockout patient-derived xenograft, and in that is this PDX1 TP3 mutant, and in another case they use TP53 wild type patient-derived xenograft. And the experimental plans is shown in this panel B, where we can see that they have different combinations. That is, in one case they use only the venetoclax, in one case they use diazoles, and there is a cases where they use both diazoles and venetoclax. So what are the results? So first we will discuss the results about this TP53 mutant. In this case, we can see that when the venetoclax is used, the effect is slightly good than what they have observed with the control. But when they use diazoles, the effects are effect of the survivals mice is much higher, and with combined therapy the effect is much more higher. And when they quantified in terms of this intratumoral biopsy of bone marrow, what they have find out is that it is minimum when this venetoclax plus diazoles is used, whereas it is maximum when the venetoclax is used. That is, it does not have too much impact. But with diazoles they see some decrease, and in the combined therapy they see a significant decrease, even significant when we compare with the results of the diazole alone. And for this TP53 wild type case, patient-derived xenograft, what they have find out is that the results are similar to the panel C, but what they have find out, the increase in the diazole treatment, or the increase in the survival rate with the diazole treatment is even around 10-fold times higher. And these things further increased when the combined therapy is used, that is venetoclax plus diazole. And these things are quantified in the panel F, where we can see that when they use venetoclax alone, they do not see too much increase, and the values are intratumoral biopsy values are almost similar to the control. But when they use diazoles, they see a significant decrease in the percentage of live cells, and which are even enhanced when they use the combined therapy, that is STING agonist and BH3 mimetics. So this all the results indicates that STING's agonist plays a role, have a role towards STING agonist have a role towards suppressing the tumor cells, or targeting the tumor cells in the AML patients, or sting expression cancer cells. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.